Dear students, now we will discuss further experiments and the work done by other scientists. As you know that the work of Garrod and Bateson that was closed until 1941 and no work was performed till then. But in 1941, these two scientists, Beadle and Tatum, Edward Tatum and George Beadle, they reopened the work and then they carried out a series of experiments at Stanford University. As they were geneticists by their profession, so they provided a very solid and very definitive evidence on the point which says that information encoded within the DNA of chromosomes, it specifies certain particular enzymes, and those enzymes are needed for proper metabolism of the body. And whenever there is a defect in the DNA or in the genetic makeup, ultimately it is going to affect the enzyme and the function it carries out. So what did they do? They took Neurospora spores. Now what is Neurospora? Neurospora is basically a fungus and its another name is pink bread mold. This is known as pink bread mold. Now the experiments were carried out in two sections. One was the control group and the other one is experimental group. Now first of all, they took the spores and they added the spores in the test tube which had the minimal medium. Now what is minimal medium? It is the minimum requirement for the growth of the spores. The minimal medium has sugar, ammonia, salts, vitamins, and this all is dissolved in water. This is minimal medium, the minimum requirement for the growth of Neurospora spores. Now when they added these spores in the minimal medium, they observed growth. It means that the spores, they grew on the minimal medium. Now they took the spores and bombarded the spores with X-rays. Now by this method, by introducing X-rays onto the spores, they were basically introducing the mutations or changes in the genetic makeup. Naturally, when the diseases were studied by Archibald Garrod and William Bateson, those diseases were caused because of the mutations that occurred naturally in the genetic or hereditary information in the ancestors of the affected families. But they introduced these mutations artificially, deliberately, just to observe how the mutations affect the organism. Now these spores, after treatment with X-rays, were again added to the minimal medium. And now there was absolutely no growth. The organisms that are treated with X-rays are known as mutants. So the mutant spores did not even grow on the minimal medium. Now let us see the next steps in the experiment.